So eukaryotes have another level of regulation that prokaryotes don't have, and that's the idea of RNA processing. Now recall that because of the lack of a true nucleus, in prokaryotes, transcription and translation have the ability to occur simultaneously. There is no need to export the mRNA into a different compartment because there is no additional compartment. Eukaryotic mRNAs, however, are different. Eukaryotic mRNAs must be exported out of the nucleus before the process of translation occurs. And it turns out that the initial form produced of the transcription event is not necessarily the final form of RNA. So what ultimately happens is transcription produces primary transcripts that then are processed into what's called mature transcripts. Now you may say this seems like a wasted step. Why not just produce mature transcripts right away? We're going to talk about the advantage of how we can maximize our genome by alternatively processing primary transcripts. Now, when I look at eukaryotic mRNA, there are two regions. There's an intron region and an exon region. An intron region contains no coding sequence. So while it's part of the primary transcript, it's not part of the final mature transcript. It is removed. Exons have the coding sequence. Exons are expressed in the mature mRNA. And the process by which we can remove introns and splice this all together is called splicing. There's other modifications, as we'll talk about, the addition of caps and tails, we'll talk about in a minute, and their important role in translation. So here it is. Here's a eukaryotic structural gene that encodes for a protein. We can see the promoter region, the terminator region, and everything in between is part of that structural component. But it doesn't mean it's all coding, and I was very careful not to say that. So we're going to transcribe the whole thing. So all the sequences that correspond to exons 1, 2, and 3, as well as the sequences that are found in introns 1 and 2, are going to ultimately produce a primary transcript. They call it a pre-mRNA. Now in the process of RNA editing, we are going to remove the introns and splice together the 3 exons. We're going to add to a 5' end a methyl cap. We're also going to add to the 3' end a poly A tail. So what is the advantage of doing these things? First of all, we can have a very diverse genome, a common set of instructions that have a very different set of answers, depending on the cell type. So in some cell types, both intron 1 and intron 2 are removed, creating for this structural gene one type of mRNA. But what would happen in another cell type if, let me get my eraser out, what would happen if only this intron is removed? Well, now if you think about that, that RNA, that mature RNA, is going to look very different and have different coding regions, ultimately producing a different type of protein. So by alternatively processing or alternatively splicing the primary transcript, we can have many different combinations. So now one gene can encode for multiple polypeptides. Sort of shooting holes in this idea of one gene, one enzyme, right? We have a genome that's about 25,000 genes, and we have a proteome, which some estimate to be 100,000 proteins. You can see based on those numbers, it can't be one gene, one protein. How can genes that number 25,000 have 4 times as many proteins that are produced? It's how we process the mRNA. So processing mRNA gives us greater diversity in our proteome. The 5' cap is going to be very important for ribosome binding. And the 3' poly A tail is going to increase the half-life, the stability of the mRNA. Let's talk about this now. We don't want the mRNA to be present at all times, because we ultimately are going to produce proteins we don't need. So at some point, the RNA has to be digested because we no longer need that protein. So what happens is enzymes called nucleases come, and I'll show it like Pac-Man, they start to chew this up at the 3' end. Well, if we just have a bunch of A's that aren't important for anything here, we can afford to lose some information, afford to lose some sequences without losing any information. So the poly A tail is going to increase the half-life of translatable mRNA. So how does splicing work? Introns are found, as we said, in many eukaryotic genes. Many of them have more than one intron, which means that we can have greater diversity in our coding region. The name of the protein complex that carries this out is called the spliceosome. The spliceosome is going to precisely remove an intron when appropriate. The spliceosome is made of proteins and also small nuclear RNAs, which help carry out the catalytic reactions of splicing.
The following animation will describe the process of RNA splicing, an important step in creating the mRNA that is involved in protein synthesis via the process of translation.
So rather than walk you through the slide and static pictures, I really think moving parts help, especially in complex processes like RNA splicing. Now, when we say creating the mRNA that's involved in protein synthesis, what we're talking about is that mature transcript. And we know there's diversity in how we can process that RNA that may result in alternative proteins being produced from a common gene depending on the cell type. So this is extremely important in creating that large proteome, which we know to be so important. All right.
RNA, possessing introns and exons, and the spliceosome. Here we see an RNA molecule with a single intron. Several signals exist within the intron that are used in the splicing process. From the 5' end of the intron, these are GU, the A branch site, a pyrimidine-rich region, and the 3' AG. The GU.
This is a common theme that we see throughout molecular biology, is that the information contained in the sequence of a molecule often plays an important role in the regulatory steps that involve that molecule. So here we have a series of sequences both at the 5' and the 3' end of the intron. Let's see here, we have a GU at the 5' end. We have that branch site,
a pyrimidine-rich region,
and
the 3' AG,
and the terminal
the
AG.
GU and AG sequences define the beginning and end of the intron. Splicing is mediated by the spliceosome, which consists of several protein RNA complexes. The first step involves two complexes that bind near the GU sequence. The RNA is then looped, and 3 other protein RNA complexes bind. This final complex then undergoes a conformational change. Introns are non-coding RNA sequences that must be removed before translation. The process of removing the intron is called splicing. The intron is then cleaved at the 5' GU sequence and forms a lariat at the A branch site. The 3' end of the intron is next cleaved at the AG sequence, and the 2 exons are ligated together.
So notice, and maybe it was subtle, this is an energy requiring process which we make cuts at both the 5' and the 3' end. The cut at the 5' end results in the formation of this lariat at the branch site. Following a pyrimidine-rich region, the spliceosome is then going to cleave at the 3' end and then causing the 2 exons that are left over to be spliced together and the intron to be recycled for its nucleotides.
As the spliced mRNA is released from the spliceosome, the intron debranches and is then degraded.
And here's a static view of what we talked about before. We have the 5', the 3' ends. We have the branch site. We make a clip at the 5' end. The lariat's formed. We then make a clip at the 3' end. That intron is then removed and recycled, and the 2 exons are then spliced together to form the coding mRNA, the mature mRNA, that's going to be the substrate for translation by ribosomes. We also have capping, and certainly there's a video you can watch on that. I'm not going to show you here in this lecture. Capping adds guanosine to the 5' end. It's involved in exporting mRNA from the nucleus, and it's also important in allowing the ribosome to know where to bind for proper translation. Poly A tail we talked about before, put 100, 200 adenine nucleotides at the end of the 3' end. It increases stability and half-life of that mRNA. As nucleases begin to chew up, they're not chewing through coding sequences, they're chewing through non-coding poly A tail, which allows us to increase the amount of time the mRNA is available for translation. And here's that picture. Here's the 7-methylguanosine cap putting on the 5' end. Here's that poly A tail on the 3' end.